My name is Samantha Bishop. I'm a master's student under the supervision of Dr. Vet Lloyd, and my job is to research the incidence rate of the zoonotic diseases Leptospirosis and Borreliosis in the Maritimes. The zoonotic diseases Leptospirosis and Borreliosis are caused globally by a range of pathogens from the bacteria group known as spirochetes. They exist in animal reservoirs and are typically transmitted to human and non-human animals through tick bites for borreliosis and saliva or urine for leptospirosis. Spirochetes can differentiate from other bacteria due to their unique shape and mobility. So first of all, leptospirosis. It's a zoonotic disease caused by the corkscrew-shaped spirochete of the Leptospira genus. This genus comprises of more than 20 species, including pathogenic, saprophytic, and intermediate categories. These are not constrained by geographical barriers. This complex and diverse genus has undergone an evolutionary period in which the genes in the saprophytic species have altered, changing some mechanisms and transformed the bacterial lineages into infectious and pathogenic serovars. Infection in mammalian species generally requires environmental exposure to the pathogenic leptospires. These typically are found in water or soil, surviving for weeks in stagnant sources or months in fresh water. The leptospires can easily enter the host system through small cuts, abrasions, or through mucous membranes. The bacteria invades the vascular system of the host, causing an inflammatory response along the way, and it eventually gets filtered out by the kidneys. The spirochetes pass through the renal tubules and are shed through the urine of the infected host, releasing back into the environment for the next unlucky creature. The infection caused by pathogenic leptospires is biphasic in humans. The acute phase begins within the first 3 to 10 days after infection, allowing the bacteria to migrate from the blood to the kidneys. It is then followed by the immune phase 7 to 14 days after the infection, during which the host's humoral immune system responds to the extracellular pathogens by developing detectable antibodies. However, the bacteria's ability to evade the host immune system is a significant contributor to the health impacts of infection. Without treatment, leptospirosis can lead to kidney damage, meningitis, liver failure, respiratory distress, or even death. Active surveillance of carrier animals can aid in controlling transmission of pathogenic leptospires. The level of endemicity of an area can be predicted based on local indicators. Direct connections have been made between flooding events and an increased clustering of dogs being diagnosed. The image here is of the St. John River, which runs through the province of New Brunswick. These two pictures are taken approximately a month apart. The left is what our river normally looks like the majority of the time, and the right is during the historic flooding event in March of 2018. Oops, apologies. As the range of infected animals shifts due to climate change and the ingression of humans, companion and agricultural animals into formal wildlife habitats increases, the spread of infection is expected to follow. Determining the prevalence of leptospirosis in the province of New Brunswick, Canada, is incredibly important to predicting and controlling future outbreaks. The other spirochete that I will be talking about is Borrelia bissettii. It's one of the 23 genospecies of the Borrelia burgdorferi sensu lato complex, which is the causative agent of Lyme disease in both human and non-human animals. This bacterium is transmitted in the eastern provinces of Canada by the black-legged tick Ixodes scapularis. It has been established in the literature that Borrelia bissettii, commonly found in the southeastern United States and Central and South America, can be carried vast distances through the travel of infected ticks hitching a ride on migrating songbirds. Because of this, there is a possibility for their introduction into novel areas. This can cause problems regarding the health of the general population on our coastal provinces, as the standard testing for Lyme disease by the Public Health Agency of Canada is to detect the presence of antibodies through the use of our two-tiered serology method in combination with patient history. And this includes details of a tick bite and recent travel specifically to areas that are known as endemic regions. Since these tests have been tailored to Borrelia burgdorferi sensu stricto, biomedical researchers have discovered that these tests may not be sensitive enough to detect other species such as Borrelia bissettii. Also, I will be talking a little bit about Borrelia burgdorferi, but as Dr. Lloyd has covered a little bit and Chris Zink will cover after me, I'm not going to get into the background here. The specific aims of my project are to determine the species in which the bacteria infect, as well as the frequency in which they are infected. In order to establish human risk of contracting these diseases provincially, we need evidence that supports the presence of the bacterium in our wildlife that serve as the zoonotic reservoirs. I will then create a geographical risk model to display the areas of high infection to determine the incidence rate of infected wildlife species. Finally, I hope to determine whether or not Borrelia bissettii and Borrelia burgdorferi share a common host species in New Brunswick. So the methods I used are fairly simple. Testing was performed on a wide range of species from various sources. Samples include the entire carcass of small mammals and birds that were donated by members of the community, and a section of tongue samples from black bears, white-tailed deer, and moose. The tongues were provided by Dr. Brian Hayden at the University of New Brunswick, who obtained the samples from local hunters across the province. Tongues specifically were used because they were the easiest to get from the hunters, as typically they were not a wanted organ. And the small wildlife samples were obtained from the Lloyd Lab Biobank, with a lot of the samples previously cataloged, dissected, and DNA extracted by Chris Zink. This collection of samples was created entirely from donations thanks to citizen scientists. So citizen science is crucial to keeping our numbers as up to date as possible, and we would not be able to do what we do without the help from our community. The extracted DNA, mainly from liver and kidneys or tongues, but not limited to brain, muscle, and skin, was amplified with nested PCR primers, using the 23S gene to determine Borrelia burgdorferi presence, the OSPA gene to determine the presence of Borrelia bissettii, which was designed by PhD student Julie Lewis from the Lloyd Lab as well, and the RRS gene coding for 16S rRNA to determine the presence of Leptospirosis or Leptospira interrogans. Also, building on the work that Chris Zink has already completed, I have used new donations and the tongue samples to continue working on updating the numbers of wildlife infected with Borrelia burgdorferi. So the numbers of samples from the large mammals were fairly proportionate to each other. I had a lot of access to those samples. However, the number of samples from the small mammals and birds were slightly more variable, as they were not as readily available or commonly found and donated by local citizens. The majority of the small mammals tested were from meadow voles, followed shortly after by deer mice, maritime shrew, and jumping mice. These 4 species make up 38% of total animals tested, and the deer, moose, and bear make up 52% of all animals tested. Out of all those tested, 11.9% were positive for Leptospira interrogans, and 3.5% tested were positive for Borrelia bissettii. As you can see here, the percent of the positive test results was fairly low for all species, but that being said, even though only 6% of meadow voles were tested positive for Leptospira interrogans, overall they were responsible for 42.2% of all the positive Leptospira test results. Again, if you look at the deer mouse, 1.5% of all deer mice tested came back positive for Borrelia bissettii, yet they held 42.1% of all positive bissettii results. Lastly, I wanted to point out that all of these samples from small mammals and birds were tested in previous years for Borrelia burgdorferi by Chris Zink, so I did not include these numbers in my results. However, as the large mammals were a recent addition to our biobank and therefore they were not previously tested, I wanted to highlight these numbers in the graph by grouping them at the end, specifically the last 3 species you see there. Moose and black bears showed to carry all 3 spirochetes, with Leptospira being the majority of their positive test results. So I have created a map to kind of show this a little bit better. So on the left is a map of New Brunswick, and it's been split into sections based on the wildlife management zones. So that shows the incidence rate of Leptospira interrogans based on the test results from white-tailed deer, moose, and black bears only. 0.4% of white-tailed deer, 2.1% of moose, and 1.1% of the black bears were positive for Leptospira interrogans. The map to the right shows the watersheds of New Brunswick, which I wanted to draw attention to since, as I previously mentioned, it has been established in the literature that leptospirosis outbreaks tend to follow periods of flooding. When comparing the 2 maps, there's a little bit of overlap between the watersheds and the areas with positive Leptospira cases. Although it is not shown well here in the Watersheds New Brunswick map, there is a river that runs through the middle of the province called the Miramichi River, and it's approximately where the wildlife zone with the highest number of cases is present. What is most surprising to me is that there are few to no cases surrounding the St. John River. This area, specifically to the southeast of Fredericton, which is highlighted in the middle there, has experienced annual flooding events, which have recently increased maximum water levels, in which 2 historic floods have occurred in 2 years back-to-back in 2018 and 2019. So again, we have the map of the province divided by the wildlife management zones with the number of positive test results for Borrelia bissettii on the left and Borrelia burgdorferi on the right. Little to no overlap of spirochete infection has appeared to occur in these zones. Overall, we do see the presence of all 3 spirochetes in our wildlife species, but it has a much higher prevalence in our small mammal and bird species. The majority of positive Borrelia bissettii tests came from deer mice. From this, we can conclude that Borrelia bissettii do share a common host species with Borrelia burgdorferi. Through nested PCR and gel electrophoresis, we have been able to detect the presence of Leptospira interrogans and Borrelia bissettii in wildlife species in New Brunswick, Canada. Although it does not appear as frequently in our large mammals, Borrelia burgdorferi has been confirmed in black bears and moose in our province. Overall, there is no denying that the bacterium is alive and well in our wildlife. The question now pertains to transmission to humans.